share
the meeting is being live streamed to YouTube. Also, there are some people attending by YouTube.
Yeah, I'll be able to share.
Sure, and they are recording. Okay, got it. Right.
Okay, can I start, Miguel?
Sure, let's go.
Thank you very much, Mike, for accepting giving a talk for us for today, for these Brazil webinars. Today we have a talk by Michael Snyder entitled Big Data and Health, as I think is the first Brazil webinar of this year. Michael Snyder is a Stanford Asherman Professor in Genetics at the Department of Genetics of Stanford University at the San Francisco Bay Area. What I can say is Michael Snyder is a very well-known scientist, and he leads a very big research group at Stanford that works on the development of tools to integrate different kinds of omics data and big data to understand human genetic variation and human health and disease. And they recently also included in the portfolio of research, of course, COVID-related issues. Mike is also a very good science communicator. He's the author of a very famous book that you can find at Amazon. It's not even expensive. Okay, it's from 2016, called Genomics and Personalized Medicines: What Everyone Needs to Know. What everyone needs to know, published by the Oxford University Press. And you can also attend a couple of TED Talks that he has on issues that are very similar to the topics he will treat today in the seminar. Okay, so you can also find Mike TED Talks at YouTube. So thank you very much, Mike. Thank you very much for accepting to give this talk. It will be great for all our students.
Great. Well, thanks very much for having me here. I'll go ahead and start sharing slides. And hopefully that's all good. Great. Well, it is a pleasure to be here and give your first seminar. I was just saying earlier, some of you have heard, sorry not to be there in person because I always have just an amazing time every time I visit Brazil. So we'll work on that in the future. So I'm going to tell you about our work today, trying to basically transform healthcare using big data. Now, I think that healthcare is broken in many different fashions. I have to confess, I don't know a whole lot about the healthcare system in Brazil, but I can tell you in the US and other places I've visited, there's basically underlying problems at many different levels. And just to illustrate a little bit of this, I can show you here some of my concerns. Basically, presently, medicine is very focused on treating people who are ill. As such, it's very reactive. When people are healthy, we typically measure very few things, and some of those have modest value, I would argue. When people are healthy, they also typically don't get measured very often, usually every few years, maybe even less. And probably one of the biggest problems is that nearly every decision about your health is based on population-based measurements. Instead, I think we would all agree it's best to be focused on keeping people healthy. As such, be very proactive. We're certainly capable of measuring many, many more things. The frequency, I would argue, depends on what you're at risk for, the frequency with which you can measure what you're at risk for and what personal trajectories look like. And perhaps most importantly, we should be very focused on individual-based medicine at a level that's much, much deeper than we're currently doing. And so we should really be focused on precision health, I would argue, rather than precision medicine. And this is a major theme of our medical school here now at Stanford. And so just to illustrate this point, it turns out, now I have this in Fahrenheit, but we're taught since we're little that your temperature when you put a thermometer in your mouth is 98.6 in the US, 37 degrees centigrade, which would be about here. Oops. sorry. Instead, it turns out really the median temperature is more like this, 97.5. But the more important point is there's a spread. This is a 25th quartile, 94.6. And here's the 75th quartile, 99.1. And so what that means in today's world is that if this is your normal baseline temperature and you go into a physician's office and they measure you at 98.6, they'll tell you you're normal, you're healthy, everything's great. You know, what are you doing here? Go home. When the reality is you're at 4 degrees Fahrenheit over your baseline, I guarantee you're not healthy. And so that's a theme I'll hit many, many times throughout the webinar. It's very, very important to understand your healthy baseline so that you can detect shifts when disease comes along. So, okay, so your health, it turns out, it's influenced by many things. It's influenced, I think we all know, by your genome, your DNA, but your lifestyle and environmental exposures can also very much influence your DNA. So pathogens, other exposures, stress, food, exercise, we know these can all impact your health. And we're in a world right now where it's possible to quantify virtually all of these, some well, some not so well. This one you can quantify now with activity monitors and such. Food is clunky and so on and so forth. But you can actually get some level. But more importantly, you can determine the effects of these different things by basically taking internal measurements on people, doing deep molecular profiling. And so a number of years ago, it's actually almost, 12 years now, we set up something we call personal omics profiling, where the idea is to collect deep data on people. We'll sequence their genome once, and then we'll do deep other kinds of measurement, epigenome, transcriptome, proteome, and such. These all are done out of people's, these are done out of peripheral blood monocyte cells, so your immune cells. Some of these are done out of same cells or plasma. And metabolomics, lipidomics is done out of plasma. And then we also take microbiome, gut, nasal, more recently tongue and skin. And more recently, we put on about 8 years ago, we started bringing smartwatches. I'll spend quite a bit of time on that today. We do deep clinical testing, questionnaires, and some advanced tests as well. So we're literally taking billions of measurements on people. And most importantly, we're also doing this longitudinally. So we'll collect samples every 3 months while people are healthy. And if an adverse event comes along, like a viral infection, we'll take many, many more samples. And we've been doing this for quite some time, median about 8.5 years on a small group of people, basically 109 people, which was meant initially as a pilot study. Now it's turned into a very, very deep data set. And those are some of the results I'll tell you about today. And you might ask, why are we doing all this? Well, we're trying to understand what it means to be healthy. How does that change over time? What happens, how does that compare between different people? What happens at the earliest time of illness? And perhaps most importantly, can these advanced technologies like genome sequencing and such be used to better manage people's health? And so with regards to this last point, can genome sequencing be used and these other technologies be used to manage people's health? The answer is yes. It turns out from these 109 people in the first 3.5 years, we had 49 major health discoveries, meaning we learned something really, really important about people's health from these deep profiles. And it's a wide range of different areas. So metabolic diseases, cardiovascular, heme oncology. And some of these were a pretty big deal. So for example, we found someone with early lymphoma, 2 people with precancerous MGUS, smoldering myeloma, 2 people with serious heart issues. And in all cases, it was pre-symptomatically. These people were not symptomatic, and we discovered this. And the other important feature is that it was no one technology that uncovered these things. Sometimes it was genome sequencing, sometimes it was imaging, sometimes it was the molecular profiling. So together, they turned out to be very, very powerful for uncovering people's underlying conditions. And again, it was all pre-symptomatic. And just to run you through a few examples, it turns out genome sequencing was important. From the first 70 people, we had 12 where we learned something pretty important from their genome sequencing. And the top 6 here, there are 2 individuals for each of these 2. These are actually what are called the American College of Medical Genetic genes. The other 6 were just things that are not as commonly looked into. These are the Mendelian traits where single mutations can put people at high risk. So the most famous one, I'm sure you're all familiar with, is BRCA1. One individual had a BRCA1 mutation, which puts them at high risk for breast cancer. This is a male, so it won't be ovarian, but they're still at high risk even for breast cancer as a male. It turns out some of these things were pretty important to know. So for example, this individual, a young person, actually had a mutation in this gene, which is involved in cardiac function. And it turns out we did a follow-up stress echo, and they actually had a bit of a concern. They actually had a defective heart, so they're actually on therapy. And it turns out their father had died of a heart attack in the 60s, and aunt had one in her 50s. So this is undoubtedly the culprit. By catching this early, this person's on drugs and is well treated. This individual actually had a mutation in this gene, which puts them at high risk for certain kinds of cancers. And one did a whole body MRI follow-up, and sure enough had early thyroid cancer, also a young person. And so that was caught, and then that was removed, and the rest is fine, so that's good. This individual we had 9 type 2 diabetics coming into the study. I'll talk about this a little more in a minute. One of them actually turns out to have a mutation in HNF1 alpha, which means they're actually not a typical type 2 diabetic. They're really a MODY diabetic, which is a very different one. They've been on the suboptimal drug for years. So the point out of all this is that genome sequencing turned out to be pretty powerful. Now, this is just the Mendelian form of genome sequencing. More recently, we've been trying to come up with new ways for analyzing genomes for complex disease. So most of you know that for most complex diseases, they're actually thought to be due to multiple variants, if you will, that put people at risk. So these are some typical examples: diabetes, coronary artery disease, AFib, so on. And the way people analyze these days is with polygenic risk scores. So these are thought to be due to common variants of very low effect, and that together, if you inherit the wrong combination, you'll be at high risk. And so people put together polygenic risk scores, and they use thousands to millions of common SNPs these days to be able to make these risk predictions. And so, and it turns out it works for the top couple percent, but it doesn't work for most people. And the reason is that there's some underlying assumptions here that probably aren't correct. First, the way this is done is they add up all these variants, that treat it like it's linear, and genetics is definitely not linear. And the second problem is it ignores all the rare variants that can be highly penetrant and contribute to these diseases. So we came up with a new way of analyzing genomes using machine learning. And we'll have, this paper is published already. We have another paper coming out in Neuron. And we applied this to a disease called abdominal aortic aneurysm. We thought this was a good one to take on because these individuals, it's a semi-common disease. It's 10% of the folks who are 65 or older, and a reasonable leading cause of death. The reason we're very interested in this is that it's usually discovered when people's aorta bursts, and then they die 90% of the time. So we think it's important to have some prediction who's going to get this ahead of time so they can be screened. And so the way we did this is a way most GWAS folks would not appreciate. We basically took a relatively small number of cases, 268 cases and 133 controls, and then did whole genome sequencing, although we only focus on the exons. And what we did was we called the pathogenic mutations in those exons. And we used a machine learning approach, added in a few other medical features, and said, together, can we find genes that are more enriched in cases relative to controls in an agnostic fashion? So it's a real machine learning approach. And what happened was the algorithm pulled out 60 genes that are highly enriched in cases relative to controls. And so I forgot to say, this is well known, AAA is well known to be a genetic disease. Very few genes have been identified prior to our work, and we wound up finding 60. So then we asked what kinds of modules are these genes in, And it turns out they're in exactly the kinds of modules you would expect: blood circulation, blood pressure, cardiomyopathy, aneurysm, so on and so forth. So we actually, the sorts of genes, I won't show you this, but we could go on and show these genes are misexpressed in both mouse models of AA and in humans who have AA. And the mouse genes themselves actually tend to be rich in heart and aneurysm kinds of phenotypes. So we think this is exactly the right set of genes that's probably underlying this disease. And so we could even go on and set up a predictive test using personal genome sequences. And it turns out if you add a little bit EHR data, you can do pretty well in a genome prediction. You might say, well, it's only a little better than EHR, but that's a big difference in terms of screening out false positives. So the bottom line is we now have a predictive test for this disease. It's normally discovered when people's aorta bursts. And so we think this can be quite powerful. And we're applying this technology to other sorts of diseases as well. It turns out it works well for ALS. It's working for, and we'll be publishing that one soon, along with COVID-19 severity. We can even predict with a machine learning approach. It's kind of cool. All right, well, genomics is really only one of the technologies that uncovered these. And it turns out imaging turned out to be powerful. We found this early lymphoma case because the person had an enlarged spleen and also had elevated certain markers. There was another young individual who actually his was picked up because he had way too much IgM relative to everyone else. So there are 1,000 samples here, and this person's 2 samples had way more IgM than everyone else, just you can see by quite a few fold. So the bottom line was that this individual we saw was something's up, did further screening, and it turns out, yeah, they have too many IgM producing cells, and it's a precancer called MGUS that can convert to aggressive cancer. So he'll get screened a lot more frequently. We had 6 people with plaques in their arteries. This is picked up by imaging as well. I'm one of the 6. This is not me, but mine looks like this. And the recommendation here is increase your statins, which is done for me, and that actually reduced my plaque. So again, this is actionable information. We're very interested in type 2 diabetes because it turns out you'll see in a minute, I'm type 2 diabetic. And so coming into the study, as I mentioned before, 9 individuals thought they were diabetic. But when we actually screened everybody, 2 more were diabetic who didn't know it. And a lot of people are pre-diabetic, and that's common. 9 out of 10 pre-diabetics have no idea. And we're very interested in how people become diabetic, and which has never really been properly studied. And it turns out during the course of the study, 9 others became clinically diagnosed as diabetic, and others had diabetic range values, and others became pre-diabetic. And so we asked, well, how do people become how do they become diabetic? And the answer is of the 9, 7 of them did not gain weight. 2 gained weight, which is a pretty typical way to get diabetic, and their microbiome diversity dropped. But the other 7 did not gain weight. And so what's going on with those 7? Well, 5 of the 7 actually just gradually became diabetic, and that's shown here for these 2. This individual was first classified, if you look at the orange line here, this is fasting glucose. Gray is actually hemoglobin A1c. So this person gradually got diabetic through their fasting glucose, and hemoglobin A1c just gradually went up. This person's OGTT was the first clue that that when they were diagnosed as diabetic. 2 other people actually seemed to spike their way there as though something triggered it. And I'm one of those 2. This is me, in fact, about 8 years of data. And so my glucose was running along just fine, but I first became diabetic after a very nasty viral infection, a respiratory syncytial virus. And it got very high, actually, after I was diagnosed. I basically cut out all exogenous sugars. I doubled my biking. I tend to bike to work, and I basically started running. And this doesn't look like much, but it's about a year of data. And about a year went by, and I brought it under control. And then it was so boringly low, I stopped looking at it. Later, someone here looked at it and said, Mike, you realize your hemoglobin A1c is 7.0 at the time, got up to 7.1. The hemoglobin A1c is a measure for type 2 diabetes. 6.5 or higher is classified as diabetic. We went back and looked, and sure enough, it turns out when it went up was when I had a second viral infection. I also stopped running, so it's not clear what triggered it, but I did start running again, and I brought my sugar levels down, and they just gradually kept going up again. So, by the way, so it's not clear whether it's running or sugar, or the viral infection, but I suspect it's a combination of both. So my glucose hemoglobin A1c and glucose just gradually kept creeping up. And finally, I actually switched as it got near 6.5 again. I switched from running to weightlifting. You can't tell, but I gained 10 pounds of muscle mass by whole body MRI. And it just, that did not work. It kept creeping up. And then finally I bit the bullet and took metformin. And it turns out I'm a non-responder. In fact, my glucose went all the way to 7.5. And so at that point, the question is, well, how do I get my glucose under control? The answer is more data. What we discovered is that we did something called glucose disposition measurements. And what we discovered is that, and from other measurements, that I'm actually insulin sensitive, and I make insulin just fine. What I don't do is release it well from the pancreas. So I have very slow glucose position. I'm down in here somewhere. And so the solution for that is a pill called rapinolide, and that actually works pretty well. It dropped my hemoglobin A1c quite a bit. And then more recently, I've switched to SGLT2 inhibitors, and that works pretty well too. So the bottom line is, this is what we call precision diabetes. By being able to better understand what was wrong with me, we could prescribe the right treatment. And we think this is very important. This can be done for everyone. One of the other things we discovered, and this is one of the most important points from the study, is that no matter how we measured people, no matter which ohm, whether it's metabolome, proteome, transcriptome, even clinical measurements or cytokines, people look more like themselves than they do like other people. And so these are just 12 people with 10 healthy visits. Every dot's a different visit, every color is a different person. And it doesn't matter which ohm or cytokine or whatever you use, people's dots cluster similar to themselves. So I'm the light blue. I have 6 years of data. My dots tend to be together. Red persons are together, yellow, brown, so on and so forth. So people look most like themselves. And that actually turns out to be fairly stable over time. Not only that, if people undergo a perturbation, such as get a viral infection or do a weight gain, weight loss, they will shift, but they still look more like themselves. This is people who did an acute bout of exercise. They ran to the VO2 max. And you can see everybody's done this. They run to the VO2 max. We sampled them across it. And they shift their profile, but they still look mostly like themselves. Again, but that's the color coding. I did it twice, and both of my sets of experiments cluster on top of each other. So the point is, these personal profiles, very, very robust, very, very stable. And so the reason we think this is important is that when you're trying to practice medicine, the best way to tell if you're getting ill is to compare a sample against your healthy baseline. That's very, very important. And this is a hard concept for GWAS people to understand. They think, oh, you need thousands of people. And that's sometimes true, but you're actually much better off, people's healthy baseline, their shift. You can't miss that change because it's usually pretty dramatic. But when you try and follow someone's healthy baseline by comparing or sick sample with that of everyone else, that's hard because the individual differences are very, very large. So if you don't remember anything else from this talk, that's the most important lesson I can tell you. Now, I told you people's profiles are pretty stable. They don't change all that much, but there are some molecules that do change. And it turns out there are seasonal changes and there are changes over time, which we're associating with aging. And so we went in and asked some very naive questions. You've been taught since you were little that there are 4 seasons, and they're winter, spring, summer, and autumn. And we can just say, well, how many seasons are there really? And you're told there are 3 months each. And if you think about it, that doesn't even make any sense. Maybe there's 15 seasons, maybe there's 3. And why would they be 3 months? Maybe some are 2 weeks, maybe some are, 4 months or 5 months. Who knows? We could let the The biological data, though, tell us how many seasons there really are. Because we had been sampling each dot's a different sample, we've been sampling across the year, we can say, well, are there seasonal patterns to the molecules we're measuring? And the answer is yes, that we found 1,192 molecules of microbes that show a seasonal pattern. And some of them were known before, like hemoglobin A1c, although we defined it at a much higher resolution. Most of them were brand new, or at least as far as we could tell. So the point out of all this is we could find lots of seasonal molecules. And so then we can next ask, well, how many patterns do these seasonal molecules fall into? Is it 5? Is it 15? And the answer is 2. And we can show that mathematically using elbow plots. And the bottom line is you say, well, that makes sense. It's California. That's where these people are sampled. One must be one must be summer and one must be winter. And there you would be half right. So it turns out a lot of the molecules peak in winter. That's these. And it turns out it's things involved in acne, viral infections, and acute responses in your immune system peak in winter. And that makes sense. That's when people often get sick. But the other was not summer at all. It turns out it was late spring, kind of April, May, early summer, late spring, early summer. And some of that made sense. Allergies and asthma were some of these molecules. But others were things like cardiovascular disease, metabolic disease, type 2 diabetes molecules. And our interpretation of this is that even in California, people don't exercise as much in winter. It does rain here and it's a little bit cold right now for us. But so we think they're less active. And so these molecules build up. And then when the weather gets warmer, they become more active and get things under control. And I should point out there's other things I don't really understand. Sleep and seizures also peak, and molecular signatures associated with those also peak around then. We think this is important information because a lot of the markers I described, they're actually medically relevant. So a lot of clinical markers show these seasonal patterns. And we think, again, knowing this could be could be very useful for guiding people's health management, knowing that they're going to hit high in some areas in April, May, and then encourage them to let it not get too high or not worrying because they're going to come out of it when May comes around. So right now, probably up in this area, these are all increasing. The other time that people's molecules change is over time, which we're interpreting as aging. Now, what's special about our study is we're following the same people over time. And basically, we had 43 people with a lot of measurements, so we could really see what was going on. And we think you need as few as 5 measurements within 2 years, and we can tell you how you're aging. And it turns out everybody's aging differently. So the one on the left, this is 4 years of data. This is me. And I'm a pretty typical ager. My coagulation, metabolic pathways all go up over time. For this person, their top pathway is cardiac hypertrophic signaling pathway. So they're a cardio ager. Their cardio pathways, top pathway going off. And we can take these 43 people and say, how many patterns do they have? And the answer is there's 4 that are statistically significant. I know they're going to be more than 4 patterns. We call these patterns ageotypes, by the way. So different people have different ageotype patterns. So, and the 4 are kidney, liver, metabolic, and immune. As I say, I know there's cardiac, but we only had 1 person there and others as well. And it turns out every column is a different person. Every row is a different ageotype. And the people down here, they're aging in all 4 categories. This person's aging in 3 of the 4. I'm somewhere in here. I'm actually a kidney, liver, and metabolic ager. But I'm not much of an immune ager, it turns out, so that's a good thing. This person's a kidney ager, this one's kidney and metabolic, and so on and so forth. So we can actually classify how people are aging. And not only that, just like the seasonal patterns, there are clinical markers associated with aging. And these are just 6 of them. I'll just show you 2 examples. One easy one to understand is hemoglobin A1c, this marker for diabetes. The group as a whole goes up. That's what this last column with the star means. If you look on average, everybody's hemoglobin goes up, and that was known already. But what's special is that if you actually look at individual cases, for these 4 people, they're going up. Dark bar means they're statistically significant. But for these 4 people, it's actually going down. And this one was very easily interpretable because what was going on here is that 2 of the people lost weight, 2 others exercised, one other exercised, 2 others went on a diet, and so on. So these folks are able to reverse this marker and their ageotype by actually a lifestyle change. Now, and this makes sense. So one would predict this. This one's a little less easy to interpret. Creatinine is a marker of kidney function. The group as a whole went up, but 10 people were statistically significant and decreasing. And so you might ask what's going on here. Well, 8 of the 10 are on statins. So somehow statins might be, helping mediate creatinine function. And that's something to follow up on. So the bottom line is, the way we think about this is that you're like a car. Everything is aging, but some parts are wearing out first, whether you're metabolic or what have you. And by knowing that, you can better adjust your lifestyle to actually try and fix those problem areas. And the other thing I'll tell you is that it does turn out everyone's aging differently. So this is using Morgan Levine's. She has something called pheno-age, an aging marker. And this is people's biological age relative to their actual age in years. And it turns out these people are aging faster than the average, and these people are aging slower. So, this is kind of what you might expect, but we can actually measure that, which is kind of cool. So one thing people ask us a lot is, how are you going to scale this? This is expensive. How are you going to scale it? And the answer is, we formed a company. This is what you do in Silicon Valley. A company's called QBio. And what they're doing is they're doing a medical version of what I just told you. And they're also doing whole body MRI. And what's happened is we've uncovered early disease in a number of different cases, all pre-symptomatically. And some of these are pretty big deals. So most were found by multiple kinds of measurements. Some were found because of longitudinal profiling. And as examples, we found one individual with early pancreatic cancer, which is almost never done. That's really, really rare. And then other kinds of cancers, fatty liver, and so on and so forth. So we found quite a bit. Now, one other area we've been pushing a lot more recently is the wearables. And we like these because you can measure people 24/7 and really get a constant measurement on people. And so we got involved in this when fitness trackers like Fitbit were just that. They were used mostly for tracking fitness. People would put them on after 3 weeks for the watch in the drawer, say, yep, I understand my pattern. Well, we realize these are pretty powerful health monitors. And in the US, they're worn by millions of people. 50 million people have a smartwatch, which is 20% of the population. And what's powerful is they make hundreds of thousands of measurements on you each and every day for the cheap ones. Some of them will make as many as 2 million measurements on you every day. So they track you in incredible depth. And they can track all kinds of different things: heart rate variability, respiration. Blood oxygen is not accurate in absolute values, but the change is pretty good, skin temperature, and so on and so forth. So we can actually track all these on people and for quite a bit. And so we're big believers these are going to be important health sensors. And in fact, I believe it so much, I use 8 of these devices every day. So if you look up, I have 4 smartwatches here. I normally wear a ring, which I've lost. I just got some recent hearing aids, and it turns out they have sensors in them too. It's pretty cool. I can track my activity with this and so on. So so I guess I have 9. That's fairly new. And so it turns out we've used these for all kinds of studies, and it's turned out they're very, very powerful for detecting infectious disease. And the way we got into this was as follows. I discovered my Lyme disease, of all things, with a smartwatch and a pulse ox. It's one of these devices that measures your blood oxygen by sticking them on your finger. And so the story behind this is I was helping my brother put up fences in rural Massachusetts, and then 2 weeks later was flying to Norway through Frankfurt. And on that last trip, I noticed that my blood oxygen was running abnormally low, around 90 median instead of 96 for that kind of a plane, that kind of a height. And I also saw that my heart rate was elevated. I later learned my skin temperature was elevated as well. This is all pre-symptomatically. And then later I did get symptoms. My temperature went up. And so I did go to a physician in Norway. I warned him it might be Lyme because of the timing, the 2 weeks Lyme disease. And sure enough, he pulls blood. Says, yep, your monocytes are up. You have a bacterial infection. He recommended penicillin. I said, no, I need doxycycline, which is what you use for Lyme disease. It was a little tense for a few moments. He did give in. It cleared it right up. You take it 2 weeks. And when I got back, I got tested. And sure enough, I was Lyme positive. Even had some antigen, but I was positive by antibody, even had antigens. And I'd given blood several days before I left and was negative. So what we think is that I seroconverted during this time. It's when the thing really took off and I got pretty ill. So the point out of all this is I was able to tell when I first got Lyme disease by simply using a smartwatch and a pulse ox. So we said, well, can we do this more generally? I had 2 years of data at the time. And we said, what can we pick up other kinds of infectious disease doing this? And the answer is yes. There were 4 times I was ill over those 2 years. One was a Lyme time. This is a viral disease, And this time I was asymptomatic. So I'm assuming it's a virus, but was never shown because it's asymptomatic. And what we discovered is that every single time I was ill every time I was ill, by resting heart rate and skin temperature, they were both elevated. So, and the Lyme ones are really, really strong. You can't miss. This is just daily summaries. You get it better even at a personal level. So the point is that we can pick up all these cases, including the asymptomatic one, with a simple smartwatch. This is all retrospective. So we wrote an algorithm we call change of heart, where we could pick up a spike in resting heart rate preceding the time symptoms came on. It was like a delta plot. And so every single time I got ill, this algorithm could pick this up. Worked on me. It worked on 3 other people, one of whom got sick twice. So every single time we could pick this up with a smartwatch. So as you might imagine, then when the pandemic hit, we published this 5 years ago now, I guess. And we were improving the algorithms, building an infrastructure I'll tell you about in a minute. And of course, along came COVID. Sorry, this is a little dated. This all looks tiny compared to what's going on now. As you know, from Omicron, the number of cases, it's record numbers. It's totally out of control in the US and most of the world too. So anyway, when this came on in March, we basically quickly put together a large team, started working on it full blast, improving the algorithms, building a data structure. And I forgot to say, how do we currently test for COVID? Well, you can do PCRs. That's the gold standard. Problem is it takes a couple of days. These days it's taking 3 days at Stanford. Very, very slow. And it's kind of expensive. There are antigen tests, which are rapid, but they're not super accurate. And then, of course, a lot of people still use thermometers, and the infrared is sort of new technology. But the problem is it's not very accurate because if it's cold outside, you don't get a good measurement. I've never seen anyone turned away with one of these infrared thermometers. So I think we do need new methods for telling when people get sick. And this is why we think smartwatches or rings can be very, very powerful for detecting when people are ill. So what we did was we wanted to see if we could use smartwatches to detect COVID. So we quickly partnered with some leading companies, launched the study. We already had it running, but had to modify it. And very quickly, we got 5,300 people enrolled. We had 32 that were what we call golden data set. These are people who were ill while wearing a smartwatch. And in this case, it was Fitbit we started with. And they had a diagnosis date and a symptom date. So they're fairly well annotated people. And we can say, well, can we detect COVID with their smartwatch? And the answer is, it turns out yes. And I'll go through this now. Here's our very first person who enrolled in the study who had COVID. And it turns out this is a resting heart rate plotted as a standard deviation plot. And this is their diagnosis day, and here's the symptom day. And it turns out that you can't miss this. The resting heart rate jumped up, in this case, 9.5 days prior to symptom onset. And we wrote a real-time detection algorithm I'll show you in a minute that works pretty good for picking this up. So a new one. And so the bottom line is that this person was probably spreading the virus for 9.5 days because they were asymptomatic and didn't know it. So for all 32 people, what we found is we could tell, see this jump up in resting heart rate in basically 26 of them, 80%. We miss it in 6 because we have a hard time getting a stable baseline, we think, for many of them. But it turns out on average, we will pick up this jump up in heart rate 4 days prior to symptom onset, 7 days prior to diagnosis. At least back then, there was always a lag, less of a lag now. It's not specific for COVID. It will find other illnesses, and this is what we would expect. Here, the median's 2 days prior to symptoms, and that makes sense because COVID has a longer pre-symptomatic period. And it's not a giant jump up in signal. The median is 7 beats per minute, but for some people, it's as little as 2 beats per minute. But you can measure that because your smartwatch is following you 24/7, making lots and lots of measurements. So we went on to write a real-time detection system for this. This is still retrospective, but basically it takes your resting heart rate on an hour-by-hour basis. It looks for a statistically significant jump up. And we've tuned the algorithm so it'll go off every 6 weeks or so. So it's reasonably sensitive. And this is a good example here. In this individual, they don't have a whole lot of background, but their alarm went off, again, all retrospective, in this case, 9 days before symptoms and 10 days before diagnosis. There's another person, I think this is 7 or 8 days ahead of time. There are other bumps. These are real signals. A lot of people get bumps over the Christmas holidays, and we know that alcohol, travel, stress can all trigger this as well. And so it could be any of these things. It could be another illness too. Here's a different illness that's non-COVID that gets picked up. And again, other signals do happen. Here is a healthy person, again, has a jump up in resting heart rate. These are real signals. But in this case, we don't know what it was causing it. So we've gone on to build a system that can scale now. We hope to do this for millions of people. I hope if any of you are wearing a smartwatch, please sign up for our study, innovations.stanford.edu/wearables. And hopefully you can send that link around. Maybe I'll put it in the chat at the end. And so we've written a new algorithm. It's a little bit lighter weight. And we're now working on a 4th generation algorithm, and it works pretty well. It's scalable to millions and millions of people. And so what we do is we set off a red alert. You actually have to click on the app, and it tells you your condition, whether you're red or green, and you'll see a red alert if you're having a statistically significant jump up in resting heart rate. And it turns out for COVID, it works 80% of the time on both symptomatic and asymptomatic cases. And this is just one of the first examples. Here's a person who got basically a red alert here. 3 days later, they got symptoms and they were diagnosed the day after that here. Okay. So again, it works 80% of the time. It also works on asymptomatic cases. Here's a person who had no symptoms. Here's their diagnosis date, presumably picked up by, random screening. And you can see they had a jump up in all these red alerts. I forgot to say we have the algorithm working for Fitbit and Apple. I'm pretty sure it'll work for anything, just so we have most of our data on these two. Here's another person, diagnosis day here, jump up in resting heart rate ahead of it. And so since we first published this study, a lot of other groups have had similar experience with smartwatches or rings for being able to detect this. We're still the only one who works on pre-symptomatic, early detection prior to symptom onset. I will say I don't think infectious disease is the only thing you can pick up with a smartwatch. We've done machine learning, and we've actually correlated other clinical markers with smartwatch features through machine learning. And basically, here's hemoglobin, red blood cell count. These have reasonable correlation. These are not accurate enough to be a predictive test, a clinical test, I should say, the clinical test, but they can give you some idea whether you're elevated for these things or not. Even hemoglobin A1c and fasting glucose, you can get a signal from a smartwatch, which is pretty cool, and some other things as well. So the bottom line is we think smartwatches, and this is an old smartwatch we use now, it's a higher generation one, we hope to do even better. I think I have a few minutes to go through a little bit of one more story, which is we also do a lot with continuous glucose monitoring. As you probably know, in the US, I'm not sure what it's like in Brazil, but 9% of the population is diabetic, 33% is pre-diabetic. So we started putting these continuous glucose monitors, they're like this, these little patches, on people who are normal or pre-diabetic just to see what their patterns look like. And what we discovered is, sure, some people had pretty good glucose control, but others are moderate and others were even severe strikers, even those who were normal by all criteria. And so we've written algorithms to try and classify people into whether they're low, medium, or high spikers. We call these glucotypes, some people running with higher levels, medium, or lower levels. And so we can classify people into glucotypes. And the other thing we've discovered is that different people spike to different foods. So some people spike to pasta, others to bananas. Some people spike to bread and peanut butter, others to protein bars. Nearly everyone spikes to cornflakes and milk. I think this is super bad. It's probably like poison, probably worse than smoking. Maybe it should be banned. But cornflakes and milk are not so great. But again, different people spike to different foods. And so we're using this to do better management of people. And as I mentioned before, some people who are normal by all measures of diabetes, fasting glucose, hemoglobin A1c, SSPG, when you actually look at them, there would be just as bad spikers as diabetics. And so we think a lot of people who have glucose dysregulation are being missed. And we think it's very, very important to pin this down. So we built a dashboard. It's called Personal Health Dashboard of My PhD. And it pulls in wearable, clinical, all kinds of data into your smartphone. We think your smartphone is going to be the most important thing in the future. By the way, I like to measure my glucose right now. Tell you what it is. 167. And the reason it's high is because I exercised this morning. I still lift weights, believe it or not. And that breaks down glycogen and spikes your glucose. So it'll come down a lot better later. But the point is you can actually track all this in real time through the My PhD app. You can share this with your physician. We think this is going to be the future of big data and health. In the future, at least of Mike Snyder's vision, I should say. I think people will be getting their genome sequenced before birth. And then together with sort of deep biochemical and omic measurements, along with smartwatches and health monitoring devices, we're going to better predict disease risk, diagnose disease early, monitor and treat disease through big data. So I've been really lucky. I have an amazing lab. I'll save a little bit of time for questions, and I'm happy to stay on a little later. But yeah, amazing lab with a talented group of folks, you, Brian, Tejas, others who have led this study. We have great clinical collaborators, Trace McLaughlin on the microbiome, George Weinstock. These are the people who built the personal health dashboard. They did the AAA. And now we have a very, very large wearable team that's actually been very active trying to build these devices so we can get genomics, get this pandemic better under control by early detection when people are getting infected. Pre-symptomatically is the goal. So that's the story. And now I'm happy to take questions. Thanks again for having me here.
Thank you very much, Mike. Terrific talk. Do you have questions? I have a couple of questions. Michelle, hi Michelle, how are you? Okay, I will read the questions.
Yeah, I see. Go ahead, fire away, Eduardo.
If earlier samples are ideal controls for composing a baseline, do you think aged individuals unaffected by a given phenotype of interest as controls may improve analytical power?
So the answer is yes, especially for themselves. So I think everybody should be getting their baseline measurements now while you're not ill. And then you will see these shifts. And I think it's especially important in the age population. I forgot to say, but the median of our population is 54. So it's an older group. And so I think it's really, really important you know your baseline. I've been in situations like this before where we have someone who's ill and we saw a disease risk. We recommended they get give samples while they're healthy, and they kind of put it off, but they're at risk for a certain disease, and then this thing hits, one we didn't understand very well. it kicked in, and we didn't have anything good to compare because we never got the healthy baseline. So I think you really need, if you want to understand a shift, you got to know what your healthy baseline's like. So absolutely, I think it's really important to get these samples and measurements, for nothing else, the samples at least, while you're healthy. So you can compare when you have an undiagnosed or some other condition to compare it to.
Michael, I also have a question. Is your cohort include several populations, ethnic groups, or is focused on a specific ethnic groups? And how this diversity, have you measured on the variability, diversity you see, how part of this diversity is due to the fact the cohort being multi-ethnic?
Yeah, it's a great question. So the answer is we're sort of typical US in the sense that we have Hispanics, we have African Americans, we have quite a few Asians, and we have more South Asians, meaning Indians and such, than a typical cohort in the US. So it is pretty diverse. We're underpowered to say if they're ethnic group specific differences, like we don't have anything that can tease that out. So yeah, so we'll just need more people. This was originally set up as a pilot study with the goal of getting millions more people. And I think we're going to get that through QBio, through this company I founded, and other means as we scale. So it will be important to see if, I think this concept when people shift up is going to be important, or shift out of their baseline for no matter who we measure. But I think being able to make better disease risk predictions from genomics and things, it will be important to have more information and set up, to some extent if possible, ethnic group specific risk predictions and things like that. So we want to get as accurate as possible in predicting health and disease outcomes.
I will read, I have another question, but I will read the other people questions.
Sure, keep firing away, and Gloria has her hand up too.
Isabella is asking, how do you integrate the epigenetics and transcriptomics like discoveries, and which type of data did you measure it?
Yeah, great question. So we are doing, we have a limited amount of whole genome bisulfite sequencing, and through the time I got diabetic, believe it or not, turns out the promoter regions of my metabolic gene shifted. Not a lot, a little bit, but it's over hundreds of genes. And we think that's probably likely what's responsible for my type 2 diabetes. And so the transcript, we see the transcript will go off every single time you have a chronic condition or illness or acute exercise. But your epigenome really, we think, only changes in response to chronic conditions like aging, diabetes, things like that. So we actually think they're measuring somewhat different things. It's a really great question you asked.
Emmanuel Diaz Neto. Hello, Emmanuel. Emmanuel is one of our Brazilian investigators on cancer, genomics of different topics. Are you working with approach to detect preneoplastic states with smartwatches, or the cancer recurrence?
Believe it or not, we are actually. I don't know if it's going to work, but we're trying to do that. We're starting with mouse models where we do xenografts or reductions of mouse tumors, and we're looking at heart rate variability, all this sort of thing. So, and we are keeping track of humans who have cancer as well, but we think we'll be a little better controlled with the mice. But the goal is to try and do both. It's a great question. Yeah, that'd be pretty wild if you can tell if somebody's getting cancer from a smartwatch. We think that'd be extremely powerful. So another great question.
Yeah. Okay, now there is a question regarding the U.S. health system and insurance regarding ethics. If someone was diagnosed with a genetic disease there in the U.S., do they have to disclose that to employers before getting insurance? This is a more general question, I think.
The answer is no, as you phrase the question, but in principle, if someone asked you and you had a clinical test with genetic information, you are supposed to disclose that to your insurer. But in the US, you're actually protected for employment and health plans from genetic information, meaning that you can't be discriminated for employment or health plans using genetic information. Now, you can still be discriminated by life insurance and long-term disability. So, yeah, I'd certainly like to see some of that change. But so you don't have to disclose, especially if it's a research study you learn the information from. So it's probably better to get it through a research study than through a clinical test, because then you would have to disclose it if they ask you.
You know, people, you can open your camera and your microphone and ask the question presently if you would like to.
Someone's asking about chronic Giovanna. Chronic kidney disease. Yeah, do you want to unmute and come on, or do you want us to read it? How do you want to handle that? And Gloria has been dying to ask a question.
Yeah,
so,
I raised my hand, but okay,
yeah, go ahead.
No problem.
Oh, who wants to go?
Giovanna, please.
Hey, how are you, Michael? Well, I'm working with chronic kidney disease. So I'm not sure if you can see me, but I think you can hear me, right?
Oh, yeah, yeah.
Well, I just wanted to know if you have any spoilers about this, if you are working with this.
I don't know about spoilers, but we are working on that, especially through the diabetes angle. We're very interested in whether we can pick up, kidney issues early. We're doing a lot of RNA-seq, and there seems to be a signal there, but we've got more work to do. But I think so, that we might be able to tell from a cell-free test chronic kidney disease. So it's something we're working on. Yeah. And I don't, we can tell the kidney age type, as I mentioned before, from the profiling as well. None of those have chronic kidney disease, though. But who knows? Maybe they'll be predictors of chronic kidney disease. Maybe we should go to Gloria. Go ahead.
I think it would be nice because, well, we have this background about how people with African ancestry, they have more, it's more likely they develop chronic kidney disease. So you have like an evolutionary factor. I'm not sure if you heard about this. I'm working with this right now. So pretty cool. So I wonder if you can do this kind of things like evaluate Asian population and African Americans. So it would be really cool.
Yeah, I would say we haven't done any predictions, especially from the genomic side. It's something of interest to us. We're busy working on some others, but yeah, no, it sounds great. Let's go to Gloria, since she's had her hand up for a long time here.
Sorry, Eduardo didn't see me. Thank you very much, Michael, for your talk. Excellent talk. I remember once you gave a talk about you following your viral, sequencing your plasma, I think, and then you detected the viruses before the infection appeared. It was fantastic. I don't know if you still are.
We're very keen on that. Yeah. And as you might imagine, with COVID, that's a big deal. Yeah. Mm-hmm.
But I have a question. It's a more philosophical question. How do you think would be difficult to translate all this information to ordinary people, especially in Brazil that rely on public health system? That's a problem because it's very, very, very interesting, but it seems to me very far from our reality.
I don't think it's that far, at least parts of it. So yes, what we're doing now is a research study, super expensive. Even QBio costs $3,500 per visit, right? So pretty expensive. They do whole body MRI. and deep medical testing. But I think once you show value, prices drop. Like we will get to $100 genome in not too long, right? And so I think that's the goal, to show this does have value. I think things that could be implemented right now, even in Brazil, is the smartwatch infectious disease. And that's going to be used for heart monitoring soon in the future and other things as well. So these devices, you know, the expensive ones are about $200, but the cheap ones, and they'll measure you 24/7, 365 days a year. The cheap ones are $50 and they still will work. So and they'll get cheaper and cheaper in the future. We're $20. And so imagine you have a health monitor for you that can follow your health all the time. That's pretty powerful. And I think anybody on the planet, that's our mission. I want to get, you may or may not realize, 3.8 billion people have a smartphone. Most of the planet, more than half the planet, has a smartphone. So if you pair that with a smartwatch, everybody can have health monitoring. And the analogy I like to use, you don't drive your car around without a dashboard. Why are we all running around without health monitoring on ourselves? You know, we count on our internal systems. They're not that good. I think these devices are way, way better. So that one I think can roll out quickly. And I think other stuff will roll out too as the price drops. That's my hope. And if we put it in the right area, we'll actually even save money rather than cost money.
Yeah, you have to give this talk to health insurance people to use that kind of.
Yeah, some have heard it. Yeah, so we'll see. The problem is the system's not incentivized. The whole healthcare system in the U.S., it's all built around illness. You go to the doctor when you're sick. You don't go to the doctor when you're healthy. And there's no financial incentive to do that. I think you should give people a discount or get employers involved, family involved. I have a whole thoughts about that. I don't know how many more questions you want to handle there.
Thank you, but
sure.
there are the hands raised.
Yeah, Jessica's had her hand up for a while. I don't know if you want to do that one next. Jessica, do you want to go next?
Yeah, please. Hi. It's a similar question, I think, that Professor Tarasona. Do you hear me? I think
we hear you, yep.
Yeah. In your data, you have 109 people, right? For your modern age and it's in other stuff. But I know it's not a big N value, but you observe some difference between ethnicity in your.
Yeah, it's very similar to the Eduardo's question. So the answer is we're underpowered for that. I don't know. We do know different individuals have different responses, but we have people, for example, doing fiber studies now, eating different fibers, and some respond to one and others respond to other. But we don't have enough power to know if any of it correlates with ethnicity. It's a great question.
Because in genetics, there's a huge lack of other ethnicity. And like you said in your early talk about the genetic polyrisk is not so efficient, but it's a personal opinion, think that's because they don't consider the ethnicity, because a lot of mutation has something associated with the ancestry. And
absolutely.
A lot of.
So I agree.
Think you will do that in the future?
I hope so. As we scale this and get more people involved. Yep, I hope so. Especially on the genomic prediction. Yeah. No, it's a great question and great point.
Thank you. It's all.
Eduardo, who's next? Should we do another hand up or?
Yeah, now people are asking. I think the Malia is want to ask. Then I think maybe Miguel, we can finish.
Hello.
Yeah.
Hello, Professor Eduardo and Professor Michael, for excellent talk. And it's a very new thing to us also, means how you are using the smartphone and other things. I have a basic question regarding COVID. You are working on the COVID for a long time, I know. But can you please give some clue why in some countries who are following high health status or different types of health consciousness, in those countries, the COVID mortality rate is very high compared to other countries? which kind of factors actually monitoring or accelerating this death rate?
Yeah, it's a great
Yes.
question. It's not clear yet, but certainly one contribution will be genetics. We've done quite a bit of genetics. We have a paper up on bioRxiv. We can show that certain alleles put you at higher risk, and it's possible that in Europeans, which, you know, there's a fair number of people get severely ill, say, compared to people in Thailand. Maybe that's the difference. Maybe it is genetics. I don't know. We'll have to see. The other possibility, you know, it's just how well you're set up to fight off an immune infection, because obviously what makes it severe is when COVID gets in your lungs. And people who have a better initial response may be able to keep it out of their lungs and not get severe COVID. And I don't know if that's previous exposures to other related viruses that's triggering that. It's not clear. It's a great question. I don't think anyone knows the answer to that.
Because I know few a group of people are working on GWAS on this particular aspect to identify the loci, and they have identified few locus also, but it is not.
Yeah, but they've only identified like I don't know, 13 genes.
Yeah,
we've identified over 1,300.
Yes. But do you think the food habit or lifestyle also playing a role in this death rate, or
I'm
any
sure that's true.
comorbidity?
Yeah, right. That's got to be true, because diabetics, obese people are much more high risk. And so for sure, running a healthy lifestyle, keeping your immune system, it's always the immune compromised people, and diabetes and obesity often compromises your immune system. So there's no question keeping a robust immune system has got to be very important.
Great. Thank you so much.
Sure.
Thank you very much. Thank you. Oh, Mike, thank you very much for your time. You for this hour of dedicated to Brazil. I think, I don't know if
I would like to. I'd like to.
Yeah, Miguel. Yeah, I don't know.
Pose a question. Michael, here in Brazil, when we want to know if you are doing okay, we go and we eat a feijoada. It's a fat dish with black beans and pork, you know. So I was wondering if you could have a selection of challenges, like having very hot Thai food, or getting drunk, or having a very cold bath, and then you do that and you inform the sensor, and then you challenge the body, so then you might see how it reacts. What about that? To have a set
we have some of that going
challenge.
on. Yeah, we have certain in our lab. We're trying different carbohydrates, for example, and black beans is one of them. So we do try different carbohydrates. I have a company, January AI, that's doing all kinds of things, just like you were describing. Look at it, exercise, things like that. So we think this is really important.
I was kind of thinking about things that our moms do not let her to do, like to go in the cold, things like that. Things that will challenge your organism, you know?
We haven't done that yet. We're interested in that, just haven't gotten to it. There's lots to do. Yeah, it's a great question. Yeah.
Sure. I think Marcelo has a question in the chat.
Yeah, I'm probably going to have to scoot off, though, for other reasons, but I'm happy to do quick questions. Let's see. Mental health conditions. We're running some mental health studies now. What's this one about there?
Okay. All right.
Yeah.
So if the audience is okay, I think we have to thank you for this very interesting opportunity to be with us. It's very important for inspiring our students, right? So thanks a lot, Mike.
Sure. Okay, very good. Thanks for having me. And yeah, good luck, everyone. Bye all.
Hope to see you in the future, but in Brazil.
Yeah, that would be great. Okay, very good. Bye.
Thank you. Thank you so much.
Okay, thank you very much.
Thank you. Bye-bye. Ok, people. Eu acho que semana que vem a gente tem um BRAZWEBINARs de novo, não sei se semana que vem ou na outra, sobre alvos de microRNA. E em fevereiro a gente tem 2 webinars com a parte estrutural, né? A gente vai ter uma modelagem de uma célula, onde o cara pega os modelos 3D e cria uma célula, e no final de fevereiro a gente tem um talk com o AlphaFold, né? Aquela iniciativa do AlphaFold no IBI, onde a gente tem 21 genomas totalmente preditos, né? As estruturas tridimensionais preditas. Então, fiquem olhando aí, só colocar BRAZWEBINARs aí no Google que vocês têm a programação, certo? Ok, Eduardo, como dizem os Teletubbies,
Ok.
hora de dar
Obrigado,
tchau.
então.
Ok.
Tchau, Glória. Você está aí ainda? Não, já foi.
Tiago está encerrando a gravação, Tiago, já, né? A
Ok.
gravação aparece no canal do YouTube, tá, gente? Quem quiser ver, vocês podem assistir, divulgar no canal do YouTube da Bioinformática de Belo Horizonte, da UFMG. Tá? Só procurar lá que todas as gravações estão lá também. Certinho, Tiago?
Exatamente, professor.
E na semana que vem, que tem outro BRAZWEBINARs ou é algum aluno?
Não, na próxima semana, dia 21, tem o Stephanie e o Pirlo. Com essa palestra.
Certo.
Das webinars.
Ok. É isso aí, gente. Sextou. Boa sexta-feira para todos aí, né? E vamos ficando por aqui. Tchau, tchau, gente.